Fusion, the international science radio show.
Ian,
Hi, Diana. How are you?
hi, nice to meet you. Good to see you.
Lovely to meet you. I had a look at your stand-up last night. Very good.
Thank you. Thanks a lot. I love your shirt, by the way.
Thank you.
So cool and spacey. Okay. So my name is Diana Robledo Ruiz. I am a research fellow at Monash University, and I do research on genetics applied to conservation.
Fantastic. So, Diana, so you're looking at genetics and the genome and the statistics and how to conserve species.
Exactly. Yes, we basically know that it's a very powerful tool to really understand what's going on with endangered species, endangered populations, and how to better help them.
Without looking at the genomics, you don't really know how healthy the populations are.
Correct. Yes. Without looking at them, you can know that, for example, they are in small numbers, that they are not reproducing as they used to. But without looking at what's inside, all their genes, you cannot know what were the processes that brought the population to that. I mean, you can see that perhaps the habitat is not doing great. But then if you don't know that they have all these genetic problems, you cannot really help them fully, because you could restore a habitat, but if the population still have these genetic problems, they won't be able to recover, and they have a very high extinction risk.
And so it's a wide variety of animals that have these genetic issues.
Yes, many. Basically, any endangered population that went from being big and connected to small and isolated, we know that they have these genetic problems, including inbreeding. So the population has gotten so small that they start mating with their relatives. And we also know that they lose genetic diversity, and that puts them in very high risk of extinction, because they don't have the genetic variation to adapt to new challenges and adapt to new problems, which is what we see everywhere with climate change.
And which species have you been focusing on in your research?
Yes, mainly my PhD, and so far in my postdoc with the helmeted honeyeater. So it's this gorgeous bird out here. It's a critically endangered, endemic to Victoria. It is actually the Victoria bird emblem, and it is gorgeous. Only 250 of them left a few years ago. I'm also starting to dab into other species like the leadbeater's possum, super cute, small possum, highly endangered as well, and the Macquarie perch, which is a fish in the Yarra River and the Murray-Darling basin that is also threatened because of human activities.
So it's not just that these populations can get so small that the only mate choices they have are their relatives. It's also that there are other diverse populations, but they can't get to them.
Yes, correct. So many times, you go from these very large populations to small islands that in the middle have not the adequate habitat. So they cannot really connect. And it is this reproduction between them, this gene flow that, brings new genetic variation, and that helps them alleviate all those problems and exchange genetic, genes, adaptations. That is pretty much the problem with also land use changes. They used to be, nice, pristine habitat, and now they are being used for farming, for agriculture, and that really puts them at risk.
Right. And I guess without the genetic variation, they can't develop resistance to diseases and changes in the environment.
Yes, correct. So we know that, yes, they don't have all those genetic tools to face changes, and changes come in many different ways. So it's always it's like a toolbox, right? The more tools you have, the more likely are you to be able to face different challenges. And then we also know that when populations get so small, they start getting more and more detrimental genes, harmful genes that start getting expressed. So they get sick, they get also less fertile and less successful at having babies. And the population is already small, already endangered, that increases their risk of extinction even more.
So what can you do to help these species be more diverse?
Yes, correct. So usually the paradigm until not long ago was to try and keep them isolated and protect them. But that's the worst thing you can do. What you really have to do is, if possible, try and bring some gene flow. So basically facilitate things so that they can mate with other individuals from other populations. There are many cases in which you have one species and then you have several populations, and there's one or maybe more that are the ones that are endangered. So, facilitating things, trying to create corridors or even in captivity to facilitate the breeding between those groups. Sometimes, however, the population you have is the last one left. So that is when you need to start exploring other alternatives, which is perhaps to mate it with a subspecies. So a very close relative, very similar. And the best thing is to choose from other sources in which you know that historically they used to have this breeding, this interbreeding, to know that you're doing everything right. You're just establishing what used to be there before we changed the environment, just reestablishing the gene flow and all the connection that was there before us, before humans.
So would there be cases where you'd also do some captive breeding?
Yes. So as you can imagine, because we are trying to change this paradigm that goes from keeping them isolated to actually trying to get them out there connected with other populations, we are very conscious that it might be not very palatable for many people. So we first do it in captivity. That was the case with the helmeted honey eater over here. We first did some genetic rescue trials in captivity, mainly thanks to Zeus victoria. So we have the helmeted honey eater and a sister subspecies, which is a very close relative. We know they used to reproduce historically. We know that they have very similar characteristics and that they are compatible. This other bird is from Gippsland. So we would bring the helmeted honey eater and Gippslanders together, and play some romantic music so that they would do their thing. And they started producing babies that are completely healthy. And actually these mixed pairs, we know now, because we proved it, they are producing even more babies than only pure helmeted honey eater pairs. And that's a great sign that this is what the population needs.
That's terrific. So for the long term, though, you'd really have to have those corridors so that they can keep on genetically mixing?
Correct, yes. So the unit of conservation is the ecosystem. And what in conservation we do is sometimes we take the sexiest species, right? The cutest, the fluffiest one, to make a case for the habitat. But yes, the priority is to heal the whole ecosystem, to heal the habitat, so that all the interactions that are within it are established and they are, healed, and all the biological processes are restored. However, we do know that you could be, as I mentioned, you could be restoring the habitat and the habitat could already be done and ready for them. But if they still have these problems, these genetic problems, they won't be able to make it. And the other way around is the truth, right? So in this case, the helmeted honey eater is getting better genetically because of all these genetic rescue trials that we've been doing. But at the same time, there is a huge amount of people and agencies that are working to restore the swampy habitat in which the helmeted honey eater lives. And they are doing amazing work restoring the vegetation, restoring the water flows, because it is a very cyclical habitat. So they have to be the both of them together. But it's correct. Long term, you need to create the conditions that they can do it naturally until the habitats are large and connected, and all these can work as it used to be.
Right. And the genomics side of this, So how do you look at the diversity of these animals?
Yes, correct. So, for example, the helmeted honeyeater is such a beloved bird that there is, a huge team of people looking after it. So we have this, amazing data set in which we have blood samples for something around 80% of the population, and we have had that data for almost 30 years. So we have 3 decades of data. We took a little bit of blood from the birds, a drop from the birds. We extract the DNA, we get the sequences, and then you are able to find, if you can imagine something like a matrix in which you have individuals in the rows, and then you have genes on the columns. You can see how much variation in each gene individuals have. And then if you look at that, comparing different populations, you can really see how one population has very little genetic variation, while others that are larger and are not facing all these extinction risk, they have much more genetic variation.
Mm.
It's actually very cool.
Can
That's
I
okay.
ask you, so is it more difficult or just very different when you're conserving the fish versus the birds?
Yes, that's a good question. Well, they are very different systems. So yes, there are different challenges to face with them. For the helmeted honeyeater, it's been such a success because in some ways it has been easy to breed them in captivity. Don't get me wrong, it's still challenging. People have been learning how to handle them, how to pair them, what they like, what they don't like, under which conditions they will breed. With the fish, it's going okay. So mainly it's about translocating some individuals from one river to another. Again, these rivers used to be connected before, but now they've gotten isolated. So transferring them. We have seen a modest amount of genetic rescues. So we've seen some interbreeding, between different populations of the Macquarie perch. However, it hasn't been as much as we would like. But knowing that hasn't been as much just gives us an idea that we can pump up those numbers to try and get them to breed more. On the other hand, we had the leadbeater's possum. So this very cute, tiny, fluffy possum. It's adorable. So leadbeater possum is super hard to breed in captivity. So my understanding is that they have not been able to breed them in captivity ever, which means that you should try and, bring individuals from one genetically diverse population to the one that is endangered. The problem is that because they are mammals, they have very complex social systems. So what happened once a couple of years ago is they tried to introduce one male into a certain population, and the male and the colony in which it was introduced, it disappeared. We don't know what happened. We don't know if it was predated or what went wrong. Then they were able to find this male in another colony, which also disappeared.
Oh.
Yes, and we don't know why. We don't know what's going on with them. So you can see that it's very tricky. You have like this diversity of some species is very easy to implement, some it's kind of okay, and in others it's very hard. But it's still, yeah, in the end you need to try to save what's left. And some people wonder if by bringing these new genes we are changing, what makes this population unique. And that is a very fair argument. We got some questions with the helmeted honeyeater asking if by introducing, genes from the Gippslander, we would be kind of erasing what makes the helmeted honeyeater unique and special. And that's a fair question. And that's something that we're looking into. We're trying to identify, like the genomic regions that make the helmeted honeyeater unique. Basically, that has, its groove, like what makes it unique. And the idea is that once we identify those ones, we should be able to track them down the line to see if they persist And also if we are getting rid of all those damaging genes, the harmful genetic variation that is affecting how they are living. So there's a lot of cool research to do with them. Every species has their challenge.
It sounds like it's a very delicate balance between intervening for the animals, well, for the species' best interests, and making sure something doesn't go terribly wrong.
Yes and no. So something that we always need to weigh, of course, there are benefits and risks of implementing genetic rescue, and that is something we are very aware of and we have always tried. There are very clear guidelines on how to avoid those risks. So that's something that we're very careful about. But we also need to remember that there are risks of doing nothing. And with these populations, we know that if we do nothing, their extinction risk is super high and we will lose them for sure. Being tomorrow, being in 5 days, being in 10 years, being in 50 years, they will go extinct. So it's striking this balance between, doing something and doing it right, and not because if you do nothing, they will for sure go extinct. So yes, there are costs and benefits of both.
Yes, and it's a serious issue. So, how did you find making it funny?
How did I find making it funny? Uh, Actually, I was very worried because, as you said, you're talking about endangered populations and climate change and how things are looking bad. But in some ways, we were told that humor is telling the truth, and fortunately, the topic lends itself kind of well to make some jokes about it, because if the population is very small, right, you start mating within relatives, and then in
Yes,
its own, it's a funny thing to think about. It's a bit yucky, which also, it allows to make some jokes out of there. And then you also get, the opportunity of telling them about this new suitor that is sexy and what is
yes,
going on there. Of course, then you have the possibility of inbreeding, mating within relatives.
yes,
Had been going on a lot in royal, in royal
yes,
settings, with royalty around the world. So that's something that you can bring also, for people to
yes.
have a reference of what can happen. So it's very nice. It was a challenge, but I enjoyed it a lot.
And how did you get involved with the comedy school for the training?
Yes, they actually sent around an email saying that there was this program to help scientists deliver their science and their topics in a funny way, in a comedic way. And it was my supervisor, the one that nudged me, I'd be like, hey, would you be interested in this because you like science communication? And yeah, it was thanks to her and to that gentle nudge that I got in.
And as a scientist, how did you find learning the rules of humour and comedy?
It was so interesting. Of course, because we are nerds, we like patterns, we like protocols, we know to know how things work and kind of like formulas. So it was super interesting to learn that there's also formulas, and patterns in humor. And yes, the instructor in the course was super knowledgeable. It was super interesting. He knew what kind of humor is preferred in different countries. So, for example,
Mm,
it's very different the humor we have in Australia versus the humor in, say, America, in the
mm.
States. So just knowing all that diversity of things and how to cater it to different audiences was really cool. Super interesting.
And I was looking at your profile and I see that you've got a Vimeo channel as well where you're putting out some science communication.
Yes, correct. I like to. So I've made this rule that every time I publish a first author paper, I make a very short video abstract in which I explain the use of it. And the idea is that it should be, for other scientists to understand what's going on in the paper easier, but also hopefully for general audiences.
I think that's so important. And I see you've done the 3-minute thesis competition as well.
Yes, that was also very fun. I participated in the School of Biological Sciences level, and I got first place on that one. So I went to the next one.
Congratulations.
Thank you so much, which was in the faculty level. And in that one, I got the runner up. And yeah, that
Fantastic.
was also very challenging. 3 minutes, trying to deliver all your message for your whole thesis of a PhD. It was challenging, but again, it's about, I guess, what I've learned is figure out what the message is, the
Yes.
main message in one sentence, and try to build around it, but focus on what you want the audience to take home from what you're
Yes.
telling them.
And it's also good to see that you're able to shorten it, but you're also able to talk at length when required. That's really good.
Yes, correct. I guess before my PhD, I was used to these more longish talks, 15 minutes, perhaps 20 minutes, in which you really it's the pyramid format in which you start with kind of the premises, small, and then you start building big until the end in which you have the last main message. And in this course, and for these smaller, shorter talks that are more for the general public, learning that it should be the other way around. So starting with this main message, the main thing, and trying to explain that thing, downwards. It's made a huge difference. And then you start noticing, yes, of course, that's what the news do. They tell you the big headline thing, what the topic is, and then they go into the details that support that main headline. I learned a lot. I
You
learned a
did.
lot. Definitely.
And are there other places we can see some of your science communication?
Well, yes. I have the Vimeo channel. I have my Twitter account, or my X account. I also did this Melbourne International Comedy Festival. And actually today in the evening, I'm going to give a talk about the helmeted honeyeater, the genetic rescue in the Pint of Science
Oh,
Festival. Yeah, I know. I've been very active lately, but you get into the mood and you pick up speed and you don't want to stop.
that's fantastic. I did the comedy thing last year with David as well and Seaton.
Ah, excellent. So something that I really liked from the course was that I was a bit worried about my accent and about me not
Yes.
being a native English speaker. But in the course, I learned that usually addressing those things not only makes a very good joke, but also kind of gets it out of the way, and then you can deliver an amazing talk. So I came with some worries, but that turned out to be, a strength. So I would encourage other people, other researchers, young researchers out there that are English is not their first language, to dive into it, and they can make something amazing, and it can be their strength.
That's really good. And you're totally right. I remember that as part of the course as well, that if you've got something there and you don't address it, the audience get distracted by that thing. And if you address it, then they move on to what you're actually saying and they listen.
Exactly. Yeah, exactly. That was a huge relief. And I also noticed that at least in our cohort, we were something like 20 people and only two of us, I think. Well, there was only two or maybe three people that we were not native English speakers.
Mm,
There might be it might be a hurdle for some
yes.
students and for some young researchers to try it out. So
Yeah.
trying to encourage them.
I think it's a really good program that David's doing. When I did my shows in Sydney last year, I had a friend come along who's done professional stand-up comedy, and he went and, told David that he's doing a wonderful job, that this is a terrific thing he's doing by teaching these things.
I agree. I agree. I think the quality of the talks and all the aspects that he teaches us, from not only script but even how to stand and how to, yeah, I thought it was really, really good. I think it's a great course as well. I was very
Yeah.
impressed.
Yeah. Yeah, same here. I'm very impressed. So hopefully I can keep interviewing the people who've gone in the course.
Yes, definitely.
um,
Yeah, I
and
think they
we
would like
can
that.
bring some attention to all these bits of stand-up. Oh, I didn't ask you if you're going to do more stand-up.
Well, not that I know of,
Okay.
but who knows? I'm open to it. Concluding statement is that there is hope, that there's out there in nature tons of populations that require genetic management, that are suffering from these genetic problems because they are very small. So they got very inbred and they're losing genetic diversity and they are losing adaptive potential, to adapt to different challenges. But there is hope that doing genetic rescue can save thousands of populations around the world, and we all get on board and understand the science behind it, and that it's genetic rescue is about reestablishing what was there before, reestablishing what the populations used to do before naturally, then we can help them for them to thrive in the years to come.
All right. Thank you so much, Diana.
Thank you so much, Ian. It was a pleasure.
Okay. Bye-bye.
Take care. Bye.
Study science, because you will find in the study of science a richer, more rewarding life.